Hi, welcome to this TEAS Science podcast with me, Tyler DeWitt. Many of the people studying for the TEAS are just so busy. They don't know where they'd find the time to sit down with a book and study for hour after hour. Well, if that sounds like you, these podcasts will help. Here, you can learn the science you need for the TEAS by just listening. Whether you're driving to work, cleaning the house, cooking dinner, working out, whatever. If you can listen, you can learn. In this episode, we're going to do an introduction to DNA, RNA, genes, and chromosomes. We'll also look at the processes of transcription and translation. These are some of the most complicated topics on the TEAS, and many people get really confused by these topics and they just kind of give up. I don't want that to be you. I think the reason why people get so confused here is because they can't see the big picture. When it comes to DNA and RNA, people usually just memorize a bunch of details and definitions with big words, and the definitions don't make any sense, but they think they need to memorize them anyway, and so the whole thing is just so confusing and demoralizing. Maybe you've already started studying these topics, and maybe you already feel confused. I want to take a different approach. Here's what I'm going to do. I want to start with a big picture. I don't want to get distracted by the small details and the confusing definitions. I want to explain the big picture of what's going on with DNA and RNA and genes and chromosomes, what they do and what they are, in simple terms you could explain to an 8-year-old. That's what we're going to do in this episode. Now, if you've listened to my other podcasts, you'll know that you can learn most of the information by just listening. Sometimes you might want to glance at a diagram, and after the episode is over, you can review with some worksheets. You can download all these resources at teasinonday.com/podcast. And if you like this podcast, if this is helpful to you, or if you have any feedback, please let me know. I love hearing from people. You can send me email at tyler@teasinonday.com. So, let's get started. We'll be talking about 3 topics here. First, the structure of DNA, what it looks like, and how it's organized in a cell. Then, we'll talk about the specific job or function of DNA, what it actually does in the cell, why it's important. And finally, we'll do a quick look at transcription and translation, which use DNA, RNA, and ribosomes. Structure and organization of DNA. So, DNA. First off, we might say that DNA is kind of like the hard drive of the cell. It contains the information needed to run the cell, or it directs the activities of the cell. You probably also know that the DNA contains information about characteristics of living things. Like, for example, the DNA has information about how you look as a person, like eye color, hair color, stuff like that. Later on in this episode, we'll look a little more closely at how DNA actually works, how it directs a cell, and how it has information about characteristics. But first, we'll talk more about what DNA is and how it's organized. In human cells, DNA is found in the nucleus, which is like a vault that keeps the DNA safe. Because it has a DNA, the nucleus is kind of like the central command center of the cell. Most people kind of know what DNA looks like, but if you need a refresher, you should glance at the diagram. There are 2 important things to know. First, DNA is like a very long thread with 2 strings or strands twisted around each other. The technical term for this is a double helix. Second, in diagrams of DNA, we usually show little sticks pointing into the middle of the spiral. There are 4 types of these sticks, and we usually abbreviate them as A, T, G, and C. These letters are part of the nucleotides that make up the DNA. Nucleotides are the building blocks that make up DNA. Super important. Let's just review all that. The DNA is made of 2 strands twisted around each other. This is called a double helix. The strands are made of nucleotides, and there are 4 types of nucleotides in DNA: A, T, G, and C. These letters are abbreviations for longer names. I'm going to say them now, but you don't need to memorize them. You just need to recognize them, and you only have to worry about the first letter. So, A is for adenine, T is for thymine, C is for cytosine, and G is for guanine. You just have to recognize those. So if you see, for example, adenine, you just have to think, okay, that's the A in DNA, adenine. Since DNA is a double helix, there are always 2 nucleotides pointing at each other on opposite strands. Look at the diagram if you can't picture what I'm talking about. Nucleotides pair up across from each other in a certain way. A and T always pair across from each other, and G and C always pair across from each other. Remember that. AT and GC. A good way to remember that is that the letters G and C look very similar. They both have a curve in them. So G and C pair, and A and T pair. Let's practice some of this stuff right now. Which of the following DNA nucleotide pairs is correct? GA, TC, AC, or AT? Well, the 2 pairs you should know are GC and AT. So, choice D is correct here. A and T pair. Which of the following nucleotides pairs with cytosine in DNA? Adenine, guanine, thymine, or uracil? Here we have the longer names instead of the abbreviations. But just worry about the first letters. Cytosine is C. What does it pair with? G. And that stands for guanine. Okay, back to DNA. You need a lot of DNA to control all the stuff going on in a cell or in a living thing. It's like you need a really big hard drive. The DNA from just one human cell is made of billions and billions of individual nucleotide letters. And if you took all the DNA from just one cell and stretched it out and put it end to end, it would be almost 6 feet long. 6 feet long of DNA for just one cell. So how do cells manage and organize and store all of that DNA? Well, in a couple ways. First, in your cells, the DNA is not just one long string. Instead, that total amount of DNA is divided into smaller pieces. And these smaller pieces are called chromosomes. I like to say that the chromosomes of DNA are like the books in a series. Maybe you're a fan of Harry Potter, and maybe on your bookshelf you've got a whole bunch of Harry Potter books lined up right next to each other. Essentially, the whole story is broken up into smaller pieces. It would be super inconvenient if you had one single Harry Potter book that contained all the stories and it was like 4 feet long, right? Instead, it's broken up into smaller volumes, into smaller books. And that's like the chromosomes. The chromosomes are the smaller, more manageable pieces or volumes of DNA. Chromosomes are like the individual Harry Potter books. Now, if the chromosomes are the individual pieces, is there a word for all of the DNA? Yes, that's the genome. The genome is all of the DNA in a cell or in a living thing. The genome is like all of the Harry Potter books together, the whole Harry Potter series or stories. And the chromosomes are the individual books in that series. In a diagram of the cell, chromosomes are often shown in shapes that look like the letter X. Chromosomes don't always look like that, but they often have that shape of an X in diagrams. Another way that the cells organize all the DNA that they have is they carefully wrap it around tiny circular proteins called histones. If you have wired headphones and you just throw them into your bag, they always get knotted up and they're impossible to untangle. So maybe you curl the wire up into a nice little circle, which can help it stay organized. When the cell wraps DNA around histones, it's basically doing just that. It's helping to keep that DNA organized by wrapping it in little circles around the histones. So, in diagrams, you'll often see the DNA neatly wrapped around lots and lots of histones. It's important to put all this together because the TEAS loves to test you on these terms and how they relate. Okay, so the total amount of DNA in a cell or in a living thing is called the genome. The total amount of DNA, that genome, is divided into smaller pieces called chromosomes. And each of those chromosomes is a long strand of DNA that might be wrapped around histone proteins. Now, those strands of DNA are made of nucleotides, A, T, C, and G. Nucleotides are the building blocks of DNA strands. So, from little to big, nucleotide, DNA strand, chromosome, genome. So, the total DNA in a cell is divided into chromosomes. But different organisms divide their DNA up in different ways. So, they have different numbers of chromosomes. In humans, we have 46 total chromosomes. 46 total chromosomes. Remember that. Humans get 23 of those chromosomes from their mother, and they get 23 of those chromosomes from their father. So they have a total of 46 chromosomes, or 23 pairs of chromosomes. This can be confusing, so here's how to think of it. Chromosome number 1. You got one from dad and one from mom. That's a pair. Chromosome number 2. You got one from mom and one from dad. There's another pair. All the way up to chromosome number 23. You got one from mom and one from dad, okay? So 46 total chromosomes or 23 pairs of chromosomes. One from mom, one from dad. It's also important to know that bacteria only have one chromosome. They only have one volume or one book of DNA. And their chromosome is circular. The DNA thread is connected into one big circle. Okay, so let's review all of this by doing some TEAS practice problems. Which of the following statements is true? Nucleotides form strands of chromosomes that make up DNA. Nucleotides form strands of DNA that make up chromosomes. Genomes make up chromosomes, which make up nucleotides, or chromosomes make up nucleotides, which make up genomes. The answer is nucleotides form strands of DNA that make up chromosomes. This is a pretty common type of TEAS question that's asking whether you understand how nucleotides, DNA strands, chromosomes, and genomes all relate. How many chromosomes are in a normal human cell? 1, 11, 23, or 46? There are 46 total chromosomes in a human cell. That might have been tricky because there are 23 pairs of chromosomes in a human cell, but that makes 46 total. Which of the following is true of DNA? It has a double helix structure. It's composed of histones. It's composed of nucleotides. It's composed of chromosomes. And it's found in the nucleus of a human cell. Well, DNA does have a double helix structure. That's the two strands wrapped around each other. DNA is not composed of histones. It's wrapped around histones, but it's not made of them. It is composed of or made of nucleotides. It is not composed of chromosomes. Instead, chromosomes are composed of DNA, and DNA is found in the nucleus of a human cell. How many chromosomes are in a normal cell of E. coli? 1, 4, 10, or 23? Okay, you might have frozen up a little bit with this question. I bet you didn't memorize how many chromosomes were in E. coli. Why would you? But you can probably figure it out. The key here is to recognize that E. coli is a type of bacteria. Remember what we said earlier? Bacteria just have one chromosome in their cells, so the answer is 1. Often, the TEAS will try to trick you like this. You'll have a question and you'll think, I can't possibly know that. I didn't memorize that. I've never seen this stuff before. But often, you can think it through and figure out the answer. Last one. How many pairs of chromosomes are there in a normal human cell? 1, 11, 23, or 46? Ooh, this one's a little tricky. Remember, there are 46 total chromosomes, but 23 pairs. So the answer is 23. 23 pairs. Function of DNA. Now that you know about the structure of DNA and how it's organized, we'll move on to looking more closely at what the DNA actually does. So far, we've been saying that it directs the activities of the cell, or that it contains information about characteristics. But for the TEAS, we need to understand more details. The stuff that DNA does looks a little different depending on your perspective. Let's say we're thinking about a whole living thing like a person. We can say that the DNA is responsible for determining their traits and characteristics. If someone has blue eyes, or they're tall, or they have curly hair, that information is found in their DNA. We can say that their DNA codes for these characteristics, or that these traits are encoded in the DNA. The information about characteristics is also passed from parents to their children. It's inherited. If we want to sound technical, we can call children offspring, which is kind of weird, but it's scientific. So the information on DNA is inherited from parents to offspring. That's why we call it hereditary material. Hereditary means something that is inherited. We can also say that DNA is genetic material, which also means it's inherited from parents to children. Now, it's one thing to say that DNA contains information about traits, or it's the heritable genetic material. But what does that actually mean for the cell? What does DNA actually do to create those characteristics? Well, from the cell's point of view, the DNA is a list of instructions that tell the cell how to make proteins and when to make proteins. Most people have never learned that. They never understood this detail. So I'm going to say it again. For the cell, the DNA is a list of instructions that tells the cell how to make proteins and when to make proteins. Be sure to memorize that. So many other concepts depend on that. Very few people understand it. For the cell, DNA is a list of instructions that tell the cell how to make proteins and when to make proteins. You might also hear the TEAS saying that DNA is responsible for directing protein synthesis. Protein synthesis is a technical way of saying making protein, and you know the TEAS always likes using complicated words and language whenever possible. Now, people often say, wait. So the DNA is instructions for making protein. How does protein in a cell have anything to do with the characteristics of a person? If the DNA is instructions for making protein, what does that have to do with eye color or hair color or anything like this? How does this make sense? Well, proteins have very important jobs. For example, I have blue eyes. And that's because there are proteins in my eyes that essentially make a blue dye that makes my eyes blue. If someone has curly hair, it's because their cells make proteins that cause the hair to be curly. If someone's cells make a lot of a protein called growth hormone, that person will be really, really tall. So all of these characteristics are caused by proteins. And the instructions for how to make those proteins are found on DNA. So, from the point of view of the whole living thing, the DNA has information about traits and characteristics. From the point of view of the cell, the DNA has information for making proteins. And these two things are related because the proteins in a cell affect the characteristics of the living thing. If someone's cells make a lot of the growth hormone protein, that person will be really tall. That's how proteins and characteristics relate. A cell makes lots and lots of proteins with different jobs. How are these instructions for proteins organized? Well, a single chromosome contains information for making many, many different proteins. It's like how a single book contains many, many chapters. These chapters are called the genes. A gene is a section of DNA that tells a cell how to make a specific protein. We can say that a gene codes for or encodes a specific protein. There are about 25,000 genes in the genome of a person, and there are many, many genes on one chromosome. A gene is a section of DNA that is the instructions for making one specific protein. Let's see how this all fits together with what we already know. Humans have 46 chromosomes. Each one of those chromosomes is a long piece of DNA. Each chromosome is made of millions and millions of nucleotides. And on each chromosome are many, many genes, which might be a couple hundred or a couple thousand nucleotides long. Those genes are the instructions that tell the cell how to make specific proteins. Each chromosome has many, many genes, but not every gene is used to make a protein in every cell. When the cell uses a gene to make a protein, we say that the cell is expressing that gene. For example, the cells on the top of your head probably make hair protein. The hair protein is expressed in those cells. But hopefully, the cells on your tongue are not making hair protein. Hopefully, hair protein is not expressed on your tongue. Genes can get turned on and turned off. Turning genes on and off is called genetic regulation. And there are certain areas of the DNA that are called regulatory genes and regulatory areas that are important for turning other genes on and off. Just remember, when you hear anything about genetic regulation, it has something to do with turning genes on and off. When a gene is turned on in a cell, it's used to make a protein, and we can say that the gene is being expressed. This stuff is really important, so we're going to do a whole bunch of practice problems. A piece of DNA that codes for a protein is best described as a genome, chromosome, gene, or nucleotide. That would be a gene. A piece of DNA that is the instructions for making a specific protein. Which of the following statements is correct? Genes are made of genomes and chromosomes. A chromosome contains many genes. Nucleotides form strings of chromosomes which make up genes. A gene contains many chromosomes. A chromosome contains many genes. Remember, from largest to smallest, it's genome, chromosome, gene, nucleotide. Choose the correct statements. A chromosome contains many genes. A gene contains many nucleotides. A gene may contain many chromosomes. A chromosome contains many nucleotides. The genome contains many genes. 4 of them are correct. The only one that is not correct is a gene may contain many chromosomes. Chromosomes are bigger than genes. One chromosome contains many genes. Which of the following is true of DNA? It is hereditary material. It is the genetic information. It directs the synthesis of proteins. It codes for physical traits of an organism. It is one of the components of chromosomes. All of them are true. And finally, which of the following best describes genetic regulation? Insulin protein is expressed in the pancreas, but not in the brain. Human growth hormone is expressed using a gene on chromosome 17. Parents have children with similar characteristics. DNA directs the expression of protein in a cell. Regulation means that genes are turned on or off. So the best example is that insulin protein is made in the pancreas, but not in the brain. The gene for insulin is normally turned on in the pancreas cells, but normally turned off in the brain cells. Keep in mind that all of these choices were true statements. Questions like this often trick people taking the TEAS because they see a statement they know is true and they just go ahead and choose it. But the question isn't asking which of these is true. It's asking which is the best example of genetic regulation. So you've really got to think carefully about what genetic regulation means and then pick the choice that's the best example of it. Transcription and translation. Finally, we're going to talk a little bit about how the cell makes proteins using the information on DNA. This is going to be a big picture view to try and prevent you from getting confused. As we've said, genes are pieces of the DNA that are instructions for making specific proteins. The DNA is in the nucleus. Now, there's a part of the cell, that's an organelle, that is dedicated to making protein. Do you remember what that is? It's the ribosome. Ribosomes are like little workers that assemble proteins. Proteins are long strings of amino acids, so the ribosomes take one amino acid after another and attach them together to make proteins. But there's a problem here. The DNA, the instructions, are in the nucleus, and they're kept safe inside it. The DNA can't leave the nucleus, and the ribosomes are out there in the cytoplasm, but they can't get into the nucleus. How do the instructions from the DNA get to the ribosomes? Well, here's what happens. If the cell needs to make a specific protein, it makes a copy of just that one gene, and that copy can leave the nucleus and go attach to a ribosome. When the cell makes a copy of a gene, it creates a short strand of something called RNA. RNA is very similar to DNA. It's made of nucleotides similar to the nucleotides in DNA. But it's only one strand. RNA is not a double helix, and it's usually very short. It's the copy of just one gene. When the cell takes a DNA gene and makes an RNA copy of it, that is called transcription. Remember that. Transcription happens when the cell copies a gene on DNA and makes a short strand of RNA. Then, that RNA copy goes out into the cytoplasm and attaches to a ribosome. The ribosome uses the information on RNA to make a particular protein. It reads down the instructions on the RNA, and it takes amino acids one after another and puts them together, eventually making a protein. When a ribosome takes the RNA and uses the instructions to assemble a protein, we call that translation. That's another important term. Translation happens when the ribosome takes the instructions on RNA and uses them to put together a protein. This RNA often has a special name. You might hear it called messenger RNA or mRNA. And that's because it's like a messenger that goes between the DNA in the nucleus and the ribosome. There are other types of RNA, but messenger RNA is a copy of a gene that tells the ribosome how to make a protein. Sometimes you'll see it called messenger RNA or mRNA. Sometimes you'll just see it called RNA. You might see any of those terms. People usually find this transcription, translation stuff really hard. So I like to use an analogy to help it make sense. I often like to compare the cell to a factory. Imagine that you have a factory with all sorts of workers making all sorts of different things. Those workers are like the ribosomes, who make all sorts of different proteins. Now, the instructions for making everything are in an office. They're kept safe in that office. We call these instructions the blueprints. The thing is, the blueprints are in a huge book or a huge binder that can't leave the office. The book, imagine it's literally chained to a desk. You can't take it out of the office. The blueprints are like the DNA, which has the instructions, but it can't leave the nucleus. How can we get the instructions from the blueprints to the workers doing the work down on the factory floor? Well, we could use a copy machine. You look up a certain product in the blueprint book, open to that page, and make a copy of that page. That copy is like the messenger RNA, and that process of copying is like transcription. Then you have an individual copy, and someone can take that copy out of the office and take it down to the workers. And the workers will use that copy to put together the products. Using the copy to put together the product is like the process of translation when the ribosomes make protein using the information on RNA. Let's review all of that one more time. The TEAS just loves to ask questions about this stuff. Then we'll do some TEAS practice problems. The DNA is like the master instruction book of all the proteins that the cell makes, but it can't leave the nucleus. The ribosomes make the protein, but the ribosomes are out in the cytoplasm. So a specific part of the DNA called a gene gets copied onto something called RNA or mRNA. And then that little piece of mRNA can go out into the cytoplasm and attach to a ribosome, and the ribosome uses the instructions on the mRNA to make protein. Make sense? That's the big picture. DNA is the giant instruction book for how to make all the proteins in a cell. The instructions for making a specific protein are called a gene. When the cell needs a protein, it copies the gene that's the instructions for the protein. The copy is a piece of messenger RNA or mRNA. That is transcription, copying DNA to make RNA. The little piece of mRNA can leave the nucleus, go into the cytoplasm, attach to a ribosome, and then the ribosome reads those instructions on the mRNA so it knows how to make the protein. That is translation. When a ribosome puts together a protein by using instructions from mRNA. So a gene on DNA gets copied to make RNA. That's transcription. Then RNA gets read by ribosomes to make protein. That's translation. Hopefully, this information is now crystal clear. Let's try a bunch of TEAS practice problems to make sure. What happens during transcription? A gene is copied to create RNA. A strand of RNA is used by a ribosome to make protein. Amino acids are combined by making peptide bonds. A strand of DNA is copied to make a new strand of DNA. A gene is copied to create RNA. That is transcription. Translation is when RNA is used by a ribosome to make protein. Which of the following is a true statement regarding a strand of RNA? It cannot leave the nucleus. It's smaller than a strand of DNA. It assembles amino acids to create protein. It's usually double-stranded. It's a copy of a gene on DNA. Two of these are correct. It's smaller than a strand of DNA, and it is a copy of a gene on DNA. You might be tempted to choose it assembles amino acids to create protein, but that is the job of the ribosome. The RNA contains the information to do that, but it's the ribosome that actually does it. What happens during translation? A gene is copied to create RNA. A strand of RNA is used to make a new strand of DNA. A strand of RNA is used by a ribosome to make protein. A strand of DNA is copied to make a new strand of DNA. A strand of RNA is used by a ribosome to make protein. Don't confuse this with transcription, which is when a gene is copied to create RNA. Which is the correct order of the following structures and processes for protein synthesis from beginning to end? Protein, ribosome, mRNA, DNA. DNA, mRNA, ribosome, protein. mRNA, DNA, ribosome, protein. DNA, ribosome, protein. Remember that protein synthesis means making proteins. We start with DNA, copy a specific gene to get a small piece of mRNA. That mRNA leaves the nucleus and attaches to a ribosome. Using the instructions on the mRNA, the ribosome makes protein. DNA, mRNA, ribosome, protein. All right, so in this podcast, we looked at an overview of DNA, RNA, genes, chromosomes, transcription, and translation. Now, when you have some time, you can download the study sheet and diagrams. You can find those resources at teasinOneday.com/podcast. And you can also find more episodes at teasinOneday.com/podcast. If you liked this podcast, if it was helpful for you, or if you have any feedback for me, please let me know. You can send me email to tyler@teasinOneday.com. Thanks for listening and best of luck getting ready for the TEAS.